Hi, I'm Jan Witkowski. I'm Director of the Banbury Centre at Cold Spring Harbour Laboratory. This is the fourth day of the 80th Symposium, the 80th Cold Spring Harbour Laboratory Symposium on Quantitative Biology. The title this year is 21st Century Genetics, Genes at Work. And I'm delighted on this glorious afternoon to have Anne Ferguson-Smith with me from the University of Cambridge, England.
Hi Anne, good to be here.
Thank you, thank you. So I usually begin by asking people to tell me a little bit about what they are going to talk about, or in your case, what you talked about.
But I did just talk about half an hour
this morning.
ago. Yes. So yes, I mean, we work on genomic imprinting, which is a really remarkable process that causes genes to be expressed from only one of the two parental chromosome homologs. And over the last, 30 years or so, we've studied imprinting and gone on these exciting journeys, actually wherever it's sort of taken us. And at the moment, one of the things we've been interested in is the mechanism of imprinting and how the epigenetic process that regulates it acts to remember the parental origin of the two parental chromosomes, and how that memory is actually maintained. And it's particularly important because during the very early embryonic development in mammals, there is, during the pre-implantation phase, a very dramatic genome-wide loss of epigenetic marks. We don't really know why that happens. If that reprogramming event was to hit imprints, it would result in the loss of the memory of the parental origin of the two chromosomes. And so we're interested in the mechanism that allows imprints to be refractory to these reprogramming events that are happening in the early embryo.
So let me take you back just one step before that. Why are there these epigenetic marks in the first place?
Right. So it's an amazing process, imprinting. So here you have two parental chromosome homologs, which to all intents and purposes have an identical DNA sequence. Mm-hmm. But imprinting is a process that turns genes on one of them and not on the other. So it has to be a non-genetic mechanism, an epigenetic mechanism. Mm-hmm. Genetics with knobs on, I call it, actually. So this is a mark that differentiates the two parental chromosomes and allows the transcriptional machinery to turn one of the alleles on while retain the other one in an off state. And this is regulated by the epigenetic machinery, initiated actually by DNA methylation that is put on the two parental chromosomes differently in the two parental germlines. So these two chromosomes come in with a methylation mark that is different on the maternally inherited chromosome from the paternally inherited one, and that is read by the transcriptional machinery differently.
Staying with this sort of very fundamental stuff for a moment, the best example is presumed X inactivation. And there it's obvious because you want to get a balance. You don't want two X's working away in the female and just one X in the male, so the female loses one X. But for autosomal genes, why does one need imprint? Why does one need to have?
So that's a very, very important question. I mean, we actually don't know why this process evolved. What is the selective advantage to turn one of your parental chromosomes, or not? It's actually not the whole chromosome. It's a region. Yeah. In the context of X inactivation, it is a whole chromosome pretty much. But in autosomal imprinting, it's relatively small subchromosomal domain. So what is the selective advantage to turn one off? And we don't know the answer to that. And there's lots of theories around about why imprinting evolved, and we could spend all day discussing that. But actually, one of the things that we've been interested in is asking whether there is what is the functional relevance to control gene dosage by this mechanism. And one of the journeys that we've gone on as we've been looking at the function of these imprinted genes is looking at their dosage in particular developmental contexts. And one interesting thing that we've learned is that many of these imprinted genes are exquisitely dosage sensitive, and that there are some instances where there is a selective absence of imprinting of these genes in particular developmental niches. And one niche that we've been looking at in particular is the adult neurogenic niche in the brain. And we've been working on a gene which has a canonically imprinted gene, which is imprinted in most places except in this adult neurogenic niche, where it turns on the other repressed allele because it needs a double dose.
Yeah.
So there's so it seems that it's quite important to be able to modulate gene dosage in this way by, and in this case it's by turning off one of the two parental chromosomes or turning it on again in a context where you need more of it.
So that was by way of sort of basic sort of helping me. But your talk this morning, you were talking about imprinting and release of marks during reproduction. I mean, during oogenesis and spermatogenesis.
So the story I was talking about today was about this very interesting factor that we identified several years ago in collaboration with John Lee and Phil Leader when John was at Harvard, a protein called ZFP57, which is a crab zinc finger protein. And there are many, many crab zinc finger proteins in the invertebrate genomes. And they seem to be quite rapidly evolving. And there's very little known about them. There's about hundreds of them, in fact. And they seem to target the epigenetic machinery and silence particular regions of the genome. In particular, they seem to have evolved to silence endogenous retroviral sequences, for example. And one of them is required for silencing imprints, for targeting repressive epigenetic states to imprints. So it raises some quite interesting issues. What is the relationship between an imprint and an endogenous retrovirus, for example? It's quite, so it's quite intriguing. We don't know the answer to that, but they do have many features in common. One of which being its ability to recruit these interesting DNA binding proteins, these crab zinc finger proteins that have the potential to recruit epigenetic states.
So this protein ZFP57 is it involved in indicating which genes need to be imprinted?
No. So actually, what it seems to be doing is maintaining a mark that is already there. So the mark comes in from the germline, and this crab zinc finger protein doesn't play a role in the establishment of that mark. What it does is it makes sure that mark is maintained during this very important pre-implantation embryo period, when all around methylation marks are being lost. It makes sure that the methylation marks are getting put back on. Yeah.
The marks get put back on or are not released in the first place?
Well, I think it's about targeting marks back on in a replication-dependent manner, although it also it looks like it may protect from active demethylation as well. So it's really we're still really trying to tease apart those kinds of details. But this protein is interesting because it binds not only in a sequence-specific manner, but in a methylation-sensitive specific manner too. So it notices when the region is methylated and binds to the methylated copy, and makes sure that after replication that methylated copy is methylated again, maintained. Yeah.
And you said this is crucial. So a knockout of this protein, does it produce embryonic lethals?
So not only is the egg has to be loaded up with this protein. So you require the protein to be present in the egg upon fertilization, and you also require the zygotic genome to be synthesizing it as well. And when you have those two things happening, you have enough to retain good, proper imprints. And when you knock out both the maternally loaded ZFP57 and the zygotic form, you end up losing your methylation memory, and the embryos die.
Is this a dose dependent thing? I mean, can you instead of knocking them out, can you have a half amount or a quarter amount?
So heterozygote mutants are all right, and in fact, if you don't have it loaded into the egg, but you do turn it on in the zygote, the animals don't die. But if you fail to have it, if you have it in the egg, but got this the right way around, it's very good. So if you fail to have it in the zygote but have it in the egg, you do die. You have a partially penetrant lethality, and you lose some of your imprints a bit. So there is a dosage effect, particularly at that very early period, and that is required for ensuring there's a proper memory. And you can lose your memory a little bit, I guess is what I'm saying, and that results in what Davor Salter today was talking about. It was a sort of chimeric epigenetic state where some cells have it and some cells don't.
Going back to the general phenomenon of imprinting, so for any given imprinted gene, is it always imprinted is gene X always found imprinted in the maternal chromosome?
Yes. So yes, that's one of its characteristic features. It's always expressed from the same parental chromosome homologue and not the other. So you always have which is different from sort of random monoallelic expression, which is what you see in exon activation, which is a random event. So you have a situation where a gene is expressed in a parental origin specific manner, and that means it is always being expressed from the paternally inherited chromosome or another one from the maternal inherited chromosome. There aren't a lot of, in fact, there are no examples that I'm aware of where you have a switch from one to the other, but there are imprinted genes that have isoforms that are expressed from one parental chromosome homologue and another isoform that is expressed from the other.
And it doesn't have any relation to, that the gene, the same gene on the two chromosomes can. No, of course it can't differ, can it?
Yeah.
No, I'm sort of struggling. Why?
So yeah. I mean, I think it's this question about. So if you're going to regulate dosage, why do you care about parental origin? If it's a mechanism to regulate dosage, why would you care about parental origin? And I think this has to be about the evolution of the mechanism, really, and trying to understand, and we still don't really, what it is about the paternal germline and the maternal germline that allows it to place methylation marks in different places that are then subsequently heritably maintained in the offspring. Right. And this may be, and we therefore have to look very carefully now about what the real differences are between maternally derived marks and paternally derived marks, and there are some patterns. So maternally derived marks are always at promoters, and the promoters, the methylation marks are always at the promoters either of the imprinted gene itself or at a long non-coding RNA that regulates a cluster of imprinted genes. The paternally derived ones are somewhere else. They're never at promoters. There's only 3 of them, and they're all intergenic. And that may be because of the timing of when these methylation marks are put on. So in the female germline, a methylation mark is put on in a growing oocyte after replication. It's post-replicative, that oocyte. You're not going to divide and make more eggs. Whereas in the male germline, that mark is placed during fetal development at a time when there's still going to be considerably more replication of these male germ cells. And DNA methylation is a mutagen, so you don't want to put methylation on at a promoter in a male germline because you can inherit a germline mutation potentially in these very important regulatory regions. That might be one reason. The other reason, which I'm going to, which is very, very speculative and there's no evidence whatsoever, is this idea that the male and the female germlines have very different agendas in their epigenetic acquisition. So, for example, when Tim Bester and Deborah Boukis knocked out the machinery that puts methylation marks at imprints on in the male germline, they ended up arresting the cells at the packaging stage. They basically couldn't do meiosis. There was a meiotic arrest. So this mechanism is required for meiosis in the male, and there's nothing more important for a male to do than meiosis. And so there was a
Oh, I
meiotic
don't know.
arrest.
I think we males have some other things that we think are rather important.
Well, it is very. That's true. But of course, it is this meiotic arrest renders the male infertile. In the female germline, that's not true. If you screw up the machinery that puts imprints on, you screw up imprints.
Yes.
So that says that the two parental genomes have different agendas in what they're doing with the epigenetic marks that are ending up regulating imprinting. And I think that is quite important that has important evolutionary implications for the process of imprinting.
You say evolutionary implications, and let's finish because we must finish in a moment. I was going to ask you, is imprinting restricted to mammals, to vertebrates, to invertebrates?
So actually, it's a very recent phenomenon that's evolved in mammals. Plants do something similar.
To silence the
well, no, it's about they have actually bona fide imprinted genes, not so many, and that they're thought to have evolved convergently quite independently from mammals, and so. So it's a mammalian-specific phenomenon, and then if you look at prototherian and metatherian and eutherian mammals, you see the acquisition of imprinting over time. So there are many imprinted genes. Well, there's a couple of hundred imprinted genes in eutherian mammals. There's none that have been identified in oviparous mammals, in platypus or the echidna. And if you look in the marsupial mammal, you have a very small subset of imprinted genes that are found in eutherians are actually found in marsupials. So imprinting has evolved, I think, at particular loci as the need has arisen over evolutionary time.
Yeah. But snakes get on quite well without any imprinting at all.
That's right, and of course many organisms can reproduce parthenogenetically, and of course. One of the reasons why you can't have parthenogenetic mammals is because of imprinting. Because you need a set of chromosomes from mum and a set of chromosomes from dad in order to get your proper transcriptional complement.
And the way we do it is much more from the parthenogenesis.
Well, that's for sure.
Thank you very much.
Thanks, Jan.
Thank you.